All right, hey everybody, welcome to our first BioLM office hours. Uh,
Woo!
we have the whole crew here today, and we're just going to talk about the main theme today is structural prediction, choosing the right model for your task. Nikhil, our fearless CEO, is going to lead us through some examples, and I think it's probably a good place to start off. Do you want to first off tell us, just for anyone not as familiar, Nikhil, like what is structural prediction in protein, design, and why is this something you might do?
Ooh, loaded question. What is structural prediction? Yeah, structural prediction is probably used in most protein engineering tasks or even DNA and RNA engineering tasks. You might work with sequences for a while when you're doing machine learning and AI, but eventually, you do want to see what the protein looks like, or the molecule looks like, how it folds. You might be doing docking to see, how well an enzyme sort of fits into its intended pocket, are the intended regions binding and binding tightly. All these things you do with protein or other molecule structure prediction. It's become pretty common to try and push the boundaries of protein structure prediction, but different models are good for different purposes and not so good for other purposes, and it might not be very obvious to everybody. And so that's one of the things that we're going to go through today. So, how do you decide what structure prediction model to use for your task?
Excellent. And before we dive in, so what kind of inputs would go into this kind of task and what do you get out of it? What kind of outputs? What do those typically look like?
Well, it depends on the model, and that's one of the comparisons that I think we'll want to note down and talk about today. Some models accept different molecules, multimodal. Some allow complexes, some allow multi-chains, some only allow single chains, and some only allow antibodies, some allow nanobodies. So, the type of model that you choose not only depends on the inputs that you have and matching up with the correct model, but also the outputs that you want and what you're trying to do. So and how accurate it is. So there's a lot of different things to think about that are kind of at cross section with each other.
Yeah, so it sounds like there's a lot of variety there. Would it make sense to like, are there families of models that would make sense to, kind of think through? Categories?
Yeah, there are, I think multimodal versus non-multimodal, and then I think other families of models are ones that just take sequence, and then others that take sequence and something like an MSA to try and improve the structure prediction, MSA and or templates like AlphaFold.
Yeah. And then maybe kind of one other question here before we dive in is, so what might you do? What are some maybe applications or what kind of comes next after you have your structural prediction?
Well, here we'll look at RMSD, which is, the task that we're kind of simulating here is you have a parent molecule and you want to look at a bunch of variants in silico and potentially see how well they bind to your target using structure complex prediction. You might want to see how well they fold because you might want to make metrics off of the protein structure prediction to help you with your downstream selection. Like for instance, does the molecules like active site or antibodies CDR region fold into the same shape as your parent molecule? Or did it change? Those things could be key for binding. So you might just look at the structure alone, even.
Excellent. Well, should we get to it?
Sure. Yeah, let's dive in. Let me share my screen.
And I believe we're going to look at a protocol here.
No, we're going to look at a notebook,
Okay.
which is not really as clean as I would like it to be, but we'll go through it nonetheless.
For the
Um, I talked about inputs and outputs and capabilities, accuracy and cost, and I don't actually have that filled out, and that's something that we can talk about and fill out, as we go through this, and we all sort of kind of look at the results. So I encourage everybody to contribute and jump in. But today, the models that we're going to look at are AlphaFold, ESM Fold, A-Body Builder 3, immune field antibody, Chi1, and Boltz-2. And I should write up here, probably the task is to compare the RMSD of predicted of a predicted IgG structure to the known structure to benchmark performance of structural prediction of each model against a hopefully a previously unseen IgG. And so, to that end, I'll just jump in and kind of show you, what we're working with and kind of get to the results, and I think it will lead to more discussion. So I found a PDB deposited in December 2023. So this is one big consideration already, when we're thinking about which model to use is, was the molecule that you're predicting the structure of in the training data set for the model, or was it not in the training data set? And, if you've got a sequence that's very close to something in the training data set, or equal to it. That's kind of nice. You'll get a pretty good structural prediction, but then you have to be more careful as you go outside the training data set. So the intention here is to find a IgG with a known structure, a crystal structure, for instance, deposited publicly after, as late as possible. I don't know when Chai and Boltz, grabbed their training data set. So this molecule might be in one of those. Perhaps even A-Body Builder 3 if it came out, I think in maybe 2025 or 2024. So, but sometimes, they're grabbing data sets from a couple of years prior. So we'll have to kind of look into that. I'm under chance, do you know off the top of your head, like any dates or
I think that for the large mainstream models like Chai and Boltz, they do a fair comparison with the older models of time of training data, but I think they continue to train afterwards on everything prior to releasing.
yeah, so we might have to get an even later molecule, deposited like, second half of 2025. But this will work for our purpose. Um, so the first thing that you can see is that there's a heavy chain and then a light chain. Um, Probably just, oops, not PDF, oh, PDB. This is the molecule that we're looking at. And here's the heavy chain, the first sequence, and the light chain. And I just extracted those out, the heavy and light. And then I used our BioLM endpoint CAD antibody to renumber the light chain first. And that gave me the CDRs and frameworks. And I just used that information to pull out, the part of that sequence we want, basically up to framework 4. So we have CDR1 all the way up to framework 4, and then did the same thing for the heavy chains. So we have our heavy chains here. And you can see that our CDR region starts at 98 and amino acid 98 and goes to 97. So if we, you counted, excuse me, 98 to 97, that would be our CDR region, and that will become relevant in a moment when we look at the structure predictions and RMSD on that region. Any comments or questions so far? Well, okay.
Yep.
So the next thing that we do here is just get the structure predictions from all those models. So first we call a BioLM class with entity equals ESM fold, and action is predict. So we're asking to do a predict, and then we're going to send a type of sequence. and we just send the heavy chain right here. And we're going to write that to disk. So those files are on disk over here so that we don't have to request those. And I'll move that out of the way for now. So next we request protein structure prediction from, I'll bump this up, from immune fold antibody. And that takes a heavy chain. So this is expecting an antibody here. That takes heavy or light. Here we just do the heavy. Then request a structure prediction from A-body builder 3. That takes both heavy and light. So we supply both heavy and light, but later we'll just pull out the heavy chain for our MST prediction. And then next we do AlphaFold. Here you can see that we're just screening with 2 of the databases, not Magnify, which is the biggest one. And then finally we take the best structure and relax it. And that's the PDB that we're grabbing from there. And then we do Chi1. The type of the sequence is protein. So this goes back to the inputs and outputs. This one can take different types of sequences, but here we're doing protein, and I just put in the heavy chain. And this is just some boilerplate code now to pull out and save the PDB. So now we have our AlphaFold PDB, which you can see here, and it's using chain A. And then we've got our Chi1 PDB, which is also using chain A. I think one of these uses chain H, I believe it might be this one. So A bodybuilder 3, just note when we do RMSD, we have to use chain H, whereas the others have predicted and called the heavy chain A. So here we're just converting the Chi1 CIF to a PDB. And we'll come back to this. Right here, I just requested the embedding or the MSAs and templates from AlphaFold so that we could theoretically input those into one of the other models. And there's one model that has pretty good RMSD that I wonder if it could even get better with the MSAs. And, we'll check that out. And that's another reason to think about using that model. So next, I'm going to move on to the actual RMSD calculations. Any comments or questions? Okay,
No.
so cool. So we have a little utility function here, thanks Chance, to compute RMSD. And so we're going to compute the RMSD root mean squared deviation, basically the error in the positions of the atoms for the protein structure predictions for the original PDB, so this one, and any of the predicted PDBs. So here the ESM fold PDB, and we're really just looking at the heavy chain. So in the original PDB, the chain is H, I think. No, there's a lot of A's. A, B, C. Oh, there's H. Okay, so it's H. And so that's why we have for PDB the parent H, and then for B, remember ESM fold makes if we predicts as chain A. Okay, so if we do an RMSD there, we get an RMSD across the full sequence of 5.75 angstroms, excuse me, which is pretty big. One sec. Okay. All right. So if we do an RMSD of the ESM fold structure prediction against the original IgG's heavy chain, we get an RMSD across the full sequence of 5.75. And yeah, that's pretty big. When we get to looking at the CDRs, you really are going to want like accuracy like below 2, even below 1 angstroms. So this is, I'd be a little starting to get a little cautious, but um, this is just showing an example of the code. We'll get to the actual analysis in a second. Um, Next, we have a function that computes RMSD on a specific region. So here, for instance, we're passing in the original PDB and ESM fold, and let me take this out of the way. And we're going to look at positions 98 through 107. So remember the CDR3 region. So now we have the RMSD in the CDR3 region, which is actually much less, than our RMSD of the rest of the chain. Okay, so we're going to fast forward and basically put all that in a table and a heat map. And I can, let's see, view and let's collapse selected code. Okay, so here we've got now the outputs. So for the full RMSD for immune fold, it's an RMSD of 1.2. You can see that compared to alpha fold and ESM fold, the RMSD across the molecule is pretty high, and it is as well with chai. But the RMSD across the full molecule is much better for immune fold and a bodybuilder 3 language. So basically, the dedicated antibody structure prediction models, while they don't take things like MSA and stuff like that, they're actually trained to predict the antibody structures much more accurately than generalized protein models such as ESM fold and AlphaFold and Chi. Part of the reason in that discrepancy is like AlphaFold wasn't trained on a lot of antibodies, whereas these others were trained on, antibody data sets. Now, for looking at the CDR3 region, which is really kind of the most important, there is something interesting. So, immune fold, the RMSD there is 1.5, which is second best, possibly we'll look at Chai in a sec. Alpha fold, pretty decent RMSD within the CDR region. ESM fold, also pretty decent. And then, interestingly enough, a bodybuilder 3 language has poor RMSD within the CDR3 region. And they do have another model, a bodybuilder 3 PLDDT, which we could check out. And I think the really interesting thing is that Chai 1 in the CDR region has an RMSD is 0.45, um, even though it has a pretty poor RMSD across the whole molecule. Um, so I don't know if Chai maybe has seen this molecule before, or perhaps we should try supplying MSAs from AlphaFold to input to Chai to see if that actually improves. But I thought this was a pretty interesting finding or value. Um, any thoughts on that? Maybe why Chai's is so good?
I think I'd like to see a superposition possibly for that prediction.
Yeah, I don't know how to do it here. Fortunately, I couldn't get Pi 3D Mall 2 to work. What do you think about the results about is this like kind of what you expected? At least as far as the full RMSD?
I would actually hope that the chai full RMSD would be a little bit better. I think that they had some good reports on their metrics for antibodies
Hmm.
as well as I'm kind of like it's interesting to me that the region RMSD for Imunefold is higher than its full RMSD. So that's kind of interesting as well.
Yeah. And I think is this one of them I had to put in heavy and light chain. I think that was a bodybuilder 3 language. Um, though we do select the chains for RMSD, I wonder if the fact that it's doing both chains makes it like a little less accurate. I don't know. So that's kind of the point of that is really Um, there are some models that are intended for certain molecules. There are some models that perform better, some models that have seen your specific proteins. And we haven't really talked about all the differences between inputs and outputs and stuff like that, but that really gets to like, okay, you really have to think about accuracy, what models are trained on, and how good are they at predicting things for your task. So let me try and put this in here, this markdown table. I don't think that's going to work. Yeah, no, it's not too much. Let's see if I can get it in. All right, what else should we talk about with respect to that?
Well, let's see. So if you're trying to kind of come up with like a bit of an algorithm for choosing your the right model, right? So I guess you have your basic modalities kind of where you start, I guess. And then
Yeah.
I suppose the what would you say next is the like you're saying the accuracy, relative to your comparison to your sequence.
Yeah. Wait, say that again?
I'm trying to assemble the steps based on, what we've been saying here. So the second step would be kind of based on, your sequence, the accuracy, right?
Yeah. Sorry. It was my microphone was getting like
No worries.
uh,
Some
so
combination of that and like, okay, what can you do with the model? What kind of inputs, outputs?
right. Yeah, I think there's do any of these provide embeddings?
Both, I think we have the pairformer embeddings for.
So that's an interesting one. You said pairformer.
Pairformer and just the sequence embeddings.
Yeah, AlphaFold you might want to use just to get the MSAs. Chai one allows putting in MSAs. Do we have that available in our API?
Yeah, MSAs for Taiwan. I'm not sure if we have templates available yet there though.
Okay.
Bolts does also have MSAs and templates though.
As input?
Yeah.
Cool. This one works. Let's see. Okay, so we got, let's try to put this together beforehand. Meanfold, does this look about right?
Yeah, I'd say one thing for the multimodals is oh, I see it there. There's ligands there too, yeah. So small molecules and ligands for bolts and Chi1 are another consideration. Bolts 2 actually gives an affinity prediction for small molecules, so that can be somewhat useful. And what's nice is that those affinity predictions have confidence metrics associated with them. So that comes kind of like out of box for just sort of seeing how confident the model itself is in that prediction.
Interesting. And Chai one has been used in is it being used in some competition for IPSE?
No, I think the adaptive competition was using bolts too, IPSE.
Okay. Um, the other consideration is when you run something like Bolt's Gen or Bindcraft is underneath the hood, those are using protein structure prediction models for ranking and getting metrics for optimization, right? So if I think Minecraft originally was using AlphaFold 2. And so you have to consider what structural model is used in these like generative ones as well. Because if we go and look at the RMSD, um, for AlphaFold for the full molecule, it's almost the highest 5.73. So um, that's another consideration is that some of these are used in other pipelines. And when you're using the pipeline, you've got to check out like, hey, which model is it using? Because that one might not be very good at structure prediction for my molecule.
Yeah, the RMSD for like the full chain is one thing they definitely use. I know Bolston uses that for filtering as well as like RMSD over like design residues. But then also like the confidence metrics. So they have filters in place there as well. And if you have a target or a framework that's very challenging for the model to model correctly, you have a lot of variability and very high confidence metrics, which don't always pass those filters. So just like sort of in consideration, like that's actually a case where if you have a template or MSA for the uh, your target and or even more potentially even better like in complex with your designed framework, then that can significantly improve those confidence metrics as well as the RMSD and the filters can still be pretty stringent, which is of
Hmm.
course better.
It might be hard to pass those in though. It often is. But I think we try and make that easy with endpoint over here, right? Getting MSAs and templates from AlphaFold, and then you can actually post those and pass those into Chai and bolts and stuff like that. Nice. As far as cost goes, I think it's pretty obvious, AlphaFold is the most expensive um, model, and then probably bolts and Chai, right? Or is it?
Yeah, I mean it kind of goes to the inputs too, like definitely running chai and bolts in single sequence mode can be pretty fast and not very expensive. But the minute you start adding MSAs and templates, or like you have higher recycles, the cost kind of increases. So that's another thing to consider too. Is especially for like antibodies and stuff. Oftentimes, they recommend higher number of recycles for like AlphaFold in some of these larger models. But that of course increases compute because that's like more iterative, you know, uh, structure prediction.
Yeah. Yeah, you're right. They can be pretty fast, but they do require more hardware.
Yeah.
If you're doing high throughput screening, these are amazingly fast. So if you're doing an antibody, and find that you need structure prediction, I mean, ImmuneFold is doing pretty well. If you can get maybe Chai RMSD around the full molecule lower by maybe providing new input or more recycles or something like that, um, that could be maybe something you could use downstream, but you definitely kind of want to choose often like a high throughput screening model that's cheaper. And then downstream, when you're selecting like, when you're going from like 40,000, you can use like ESM fold or immune fold. And then when you go, you need to select like your final 96 sequences or 10 sequences, you want to maybe a little more expensive and more accurate structure prediction model. And then maybe, there you want to take the final sequences and like predict them with Chai using a template or MSA as input. Um, I mean, they all pretty much scale on cost for length. Um, I don't think that's avoidable for any of them.
One other thing, just to sort of mention, I think Andrew had asked earlier, like how is this useful in like protein design campaigns? Well, so there's still a lot of classical methods involving like MD or potential structural based scoring uh, that these can actually significantly enhance just like starting points. So in terms of like the number of time steps you might need to do if you have an existing structure already. that can kind of speed things up there. So when you're talking like expensive downstream, steps, and accuracy, well, that kind of goes for the more classical non-ML pipelines as well. Like, screen really quickly with the ML and then pass it off to a more expensive ML and then potentially take those expensive structures and do an even more expensive calculation, which can
Yeah.
significantly reduce your cost and speed up compute. Like a good example of like sort of the same paradigm is how AlphaFold uh, usually uses a fast relax protocol on the predicted structures afterwards. Um, so that's like kind of like the downstream steps that can kind of enhance the efficacy of these models too.
Yeah.
So with so many new models coming out and variants, all the time as well, what would you look for as far as, when we're thinking about like model selection here, what are kind of aspects of new models or new iterations coming out that you'd be taking into consideration?
I think with the multimodal sort of paradigm becoming super favored, just like looking for a performance increase along all of those, especially in some of the harder to model tasks like antibodies or RNA sequences. There's a lot of models too with very nice papers where they have very good benchmarks, and that's always something I like to look for too. It's just like how good did they benchmark it? Do they take into consideration, like, as Nikhil was saying, the time frame of their like training test sets to make sure there's no data leakage, and as well, I think like just having a good breakdown on just each of the different areas that they excel in is really good.
Awesome. All right. Any other questions? Any other discussion we want to go through? Anything else we want to show?
I think out of this is probably, I'll clean this up and make some tables. I think there's a good blog post
Mhm.
with, pulling out some of these tables and discussing some of this stuff and can even, I think, have some clips of this throughout the blog post, like a or, like a larger in the blog post have, video itself, right? Why not? Um, I'm pulling out some other tables of, I think for these, I think one of these sections, and then I think we should totally, yeah, turn that into some content.
Yep, absolutely. And then this video should also end up in the BioLM Academy on YouTube. So,
Yeah,
Sure. One other thing like we can iterate in the blog post that's like my favorite thing to talk about with the structural prediction models is like why like the, one like Nobel and all this stuff was you know um like the dark proteome stuff, like in terms of like druggable targets and a lot of this stuff, like these structural models really kind of enabled like just um, you know, exploration of some of these targets and unknown, sequences and stuff that like, I don't know, it's just really exciting for like both, the scientist and like the farmer person to be like, hey, I can actually do these campaigns or choose, some of these previously unknown, um, you know, uh,
structure.
you know,
Yeah,
targets. It's like super cool, right? Couldn't do that before. And
totally.
how expensive, doing the actual putting a structure in the PDB has gotten like a lot easier, but it's still pretty expensive. So yeah, it's pretty nice.
Yeah, it's pretty cool. You have a molecule and can get a probably pretty accurate structure prediction. Be like, oh, does this maybe bind to something? Okay, maybe I'll test it. Yeah, or maybe not. Yeah. Um, cool.
yeah. All right. Thanks so much, Nikhil, for sharing all that. And thanks for everyone's input.
Thanks.
Should we take a stab at, any suggestions just from this group as to what we might want to tackle as a main theme for the next office hours? And put it to the community as well. But
Um, I think we could go down again a little more on the antibody stuff. So maybe we could introduce some of the uh, antibody language models we have for potentially assessing like naturalness or some of these other scores. Uh, as well as like maybe like proxies you might want to use, just even like regex wise on your designed antibody sequences to remove ones with liabilities.
all right. Thanks, everyone. Thanks, Nikhil, for walking us through that and Chance and Ahmad for answering all the questions. And yeah, we'll do this again pretty soon. So thank you.
Thanks.
Thanks. Bye.